As we start to look at interphase, or excuse me, outside of interphase, and we go into mitotic division, let's quickly review what a chromosome looks like. Some organisms have 2 of these, some organisms have more than 2. When the chromosome is doubled and we're at this stage and we have our centromere with its kinetochore, we officially call this a doubled chromosome, if you will. But even more importantly, I want to refer to each side of this chromosome as a chromatid. Okay? Don't get the vocabulary confused. Remember, before it was a chromosome, it was chromatin. Chromatin doubled to give you chromosomes. But we have to refer to each side of these as something, so we're going to call each of these a chromatid, and in fact, they are going to be called sister chromatids. That just makes the explanation and the understanding that much easier, because now we can refer to them as something. And as you know, these are eventually going to split and graduate and become chromosomes, but let's not get ahead of ourselves. Okay, let's talk mitosis. Mitosis is going to be divided into literally 5 phases. Let's list the 5 phases. First of all, we've already talked about interphase, and that's not officially a phase of cell division. But we're going to go into something called prophase, prometaphase, and you know, it really becomes a question of what decade are you learning this in? Because we used to take prophase and divide it up into early, middle, and late prophase, and then go right into this next phase called metaphase. And there's a reason I'm stressing this to you. There is no such thing as phases in the sense that, okay, time to stop one and move on to the other. This is a dynamic, continuous, very gradual process. But once again, for the sake of discussion and publication and communication, we have to call these things something. So when I say a metaphase chromosome to you, you got something pictured in your head, as opposed to a prophase chromosome. So we need to have these words. After metaphase, we're going to leave metaphase and go into anaphase. And then our last phase will be telophase at the end. So what we have, therefore, is this division of time. It's a division over time that we're going to look at this. Okay, so let's go through it a phase at a time. Let's take a look at the vocabulary, but most importantly, what do we want? We want to see the events. All right. First of all, what happened in interphase? Interphase GS1, G2, the cell got ready for this, and here's the thing. Early in interphase, the only thing, well, everything is important, but the one thing that's going to affect us is that area called the centrosome. In interphase, you guys recall that there is a structure in cells called the centrosome. And in animal cells, within the centrosome are these structures called centrioles. Early in interphase, the centrosome region doubles. So you get 2 of them. Okay? And indeed, in animals, the centrioles also double. The centrioles are kind of a puzzle for us. You know, if you remove them, like if you laser beam them, mitosis just works fine. So you know, and plants don't even have centrioles. So they're certainly associated with the centrosome, but the centrosome is the business end of what's going to happen in mitosis. Okay, so what do we have here? Well, right here we have what typically I'm going to refer to as an interphase cell. Let's review some cell plumbing here, if you will. Alrighty, so we got the centrosomes and centrioles. All right, let's move this up a wee bit and take a look at some other parts. Okay, remember the nucleus, and you know that you have within the nucleus chromatin, which has not, as we enter mitosis, it's doubled, but has not necessarily started its supercoiling. We have the cell membrane, which I'll call CM, okay? We have the nuclear envelope, which I'll call NE, for lack of space and time. And we have this structure right up here called the nucleolus, which I want to write that down for you in case you don't remember that name, nucleolus. And if you're from another part of the country, it's the nucleolus, but you got it. All right, let's take a look at what happens as we move from interphase to prophase. Okay? As we move from interphase to prophase, you notice that what begins to occur here is the centrosomes are starting to separate. Okay, they're starting to move, and it looks like they're starting to grow some fibers out of them. And in fact, we call those asters. Aster as in star. Okay, so we have the beginning of asters, and we will see in just a bit that the asters are literally microtubules that are going to make up, well, we'll give you names in a minute, but they're microtubules, suffice it to say. Look what's happening to our chromatin. It's already starting to look like that chromosome I showed you a second ago, with its centriole with its centromere in the center. So what we have here, therefore, is right here. These two lines are denoting sister chromatids, and of course we have a doubled chromosome. All right, that's nice. But what does this really look like in a cell? Well, let's take a look at a cell. If I can get this paper off here. There we go. All right, let's take a look at a cell. Here is a typical animal cell in interphase. Okay, you can see the centrosome, you can see the nucleus, you can see the dark staining chromatin, and you know, as I look at this, if I'm looking at it through a microscope, I'm saying, wow, it looks like it maybe has started to enter prophase, because I'm starting to see not the chromatin stained as an amorphous blob, but it's starting to look like the chromatin is starting to supercoil and perhaps form chromosomes. And so we start to get into prophase, where the chromosomes begin to form. And look at this, you guys. Look at this. We have the centrosomes have started to come across, and they've started to migrate. And in fact, this is well advanced. And the asters are radiating out. So this is prophase as we begin to enter the next phase, which is going to be prometaphase and eventually into metaphase. Let's take a look at that. In prometaphase, something starts to happen to the nucleus. The nuclear membrane starts to break down, the nuclear envelope. So the nuclear envelope breaks down, and we've denoted this with these chunks, okay, of nuclear envelope, which allows these spindle fibers that are growing to literally start to penetrate into the what used to be the nucleus. So in prometaphase now, we're getting the breakdown of the nucleus, so fragments of the nuclear envelope. Look at our centriole, our centrosomes. They're at either pole, if you will, and you have the chromosomes as clearly doubled sets of chromatids. Interestingly enough, at this point, look what's happened. Some of the fibers have attached to the kinetochore. And so we have here, I'm going to say that this is a kinetochore, kinetochore fiber, or tubule, or fiber, whatever. And here's, oh look, here's some tubules or fibers that have not attached to kinetochores. They may end up kind of important in a little bit too. So we have what I'm going to refer to as non-kinetochore tubules, okay, or filaments, well, not filaments, or fibers. And oh, there's my centrosome at either end. And then as we enter metaphase, very cool thing begins. That tug of war I showed you before. And by the time we're at metaphase, we're ready to start dividing, because look what's happened, very simply. We have lined all of our chromosomes. The chromosomes are as tightly packed as they're ever going to get, and we have lined all of our chromosomes on what is now seems to be a spindle. See, it looks kind of like a spindle, if you know what a spindle is, okay? And they're called spindle fibers. Some of these fibers go across the cell and don't attach to a chromosome, and some are directly attached to the kinetochores of the chromosome. And in fact, we refer to this so-called equator, or this imaginary line across there, where the centromeres indeed are lined up. We call that the metaphase plate. Okay, so that's going to be called the metaphase plate, that imaginary line, if you will, where they're all lined up. Well, what does this really look like? Glad you asked. Let's take a look at a cell in metaphase. How nice. There's my cell. There's my two centrosomes. There's my spindles, and there's my chromosomes lined up on the metaphase plate. Oh, beautiful. When you see this stuff through a microscope, it's downright exciting. All right, well, we're almost done because now we get to go to anaphase. In anaphase, the cell begins to pinch inward, okay? And we start to get the cleaving of the cell. And let's see what's going to happen here. The cell begins to elongate. This is anaphase. All right, and finally we're going to reach something, well, eventually we'll get to a phase called telophase where we're going to pinch that thing, but not completely apart. But let's look at anaphase. Very simply, what has begun to happen is that reeling in process I told you about. The chromosomes, like little Pac-Men, the motor molecules along the kinetochore. Remember, there's a kinetochore on each chromatid. The little Pac-Men along the kinetochore just are going to eat their way along that fiber and look like they're getting reeled in. So if we look at it with this chromosome here, this is going to separate and start to pull like that. Which explains, look, it explains the way I have these chromosomes arranged. Let me just show you this real quick. This is so cool. All right, to put it like I had it right there, they start to work their way in, and look at the directions the chromatids point because of the forces. And indeed, that's exactly how we've drawn it for you. So in anaphase, they start to pull in, and then what's going to happen is you're literally going to start elongating the cell. The cell elongates, and that's because of the non-kinetochore microtubules that are literally kind of pushing that cell. By pushing on the middle, the cell is going to be pushed inward. So it's elongating, okay? So the cell begins to elongate, and we finally get to telophase. But before we get to telophase, let me just show you one more thing. Let me show you what anaphase looks like in a cell. See, they start to pull inward, like so. And here's a later anaphase right here, but I have a close-up of that cell. Here's another early anaphase right there. But look at this cell. Let's look at this bottom cell. Look at that, how they're pulling all the way across. And now finally, we get to telophase. And in telophase, we start to form something called the cleavage furrow. And in the cleavage furrow, the cell begins to cleave. It begins to break. And so, break's not a good word. It begins to divide. And we're getting ready for the next step, the final step, which we'll talk about later, called cytokinesis. But look what starts to happen in telophase. In telophase, we're going to start to get the reforming of the nuclear envelope. See? And that's happening right here. The nuclear envelope begins to reform. Look, the centrosomes are divided. Look, we're starting to get the division of a cell right here. Look, oh, what's that? My gosh, the nucleolus is reforming. What are we getting, you guys? We're getting a new cell. But what have you accomplished? In summary, what have you accomplished? You have taken these chromosomes and literally split them so that now no longer are they chromatids of a pair, but they are now in separate cells. One. And a new nucleus has formed around them. One. Trust me, a new nucleus will form around this. 2, a new nucleolus appears. And what can these cells do? Well, we're going to see next how we got to these 2 separate cells from that one, and that process is called cytokinesis. But look at what you've done. You've doubled, you split. The concept is there. We need the names so we can describe it.